Have you ever wondered how genome editing works? It's an intricate process that enables scientists to alter an organism's genetic code. To do this, they use a variety of tools, all of which involve enzymes that act on DNA. These enzymes are known as nucleases. Nucleases play a crucial role in the world of genome editing. They are akin to molecular scissors, cutting the DNA strands at precise locations. Now, there are a number of different tools available for gene editing. Each of these tools has a way to guide these nucleases to specific target sites within the genome. Whether it's a guide sequence or specific DNA binding domains within the nucleus itself, the principle remains the same. The nucleus is directed to the target DNA, where it can then take action. So, the tools for genome editing all work on a similar basic principle. They direct certain enzymes to specific target sites in the genome. Now, how exactly does this genome editing process work? Let's dive right into it. At the heart of genome editing is the magic of enzymes, specifically nucleases. These are like the molecular scissors of the DNA world, designed to recognize specific sequences within the vast genetic code. They're guided to their targets by either a guide sequence or DNA binding domains within the nucleus itself. Once they bind to the target DNA, they're ready to do their job. The process of editing can happen through several pathways. Let's talk about two major ones: non-homologous end joining, or NHEJ for short, and homology-directed repair, which we'll call HDR. Imagine you're trying to fix a broken piece of string. One way to do it would be to simply tie the two ends together. That's a bit like NHEJ. The nucleus creates what we call double-stranded breaks, and the cell tries to fix the break by just joining the ends together. But this method is a bit of a wild card. It's error-prone and often results in insertion and deletion mutations, or indels. These indels can cause the gene to become non-functional, a process we call gene knockout. Now, let's look at the second pathway, HDR. Suppose you have a guide on how the string should be fixed. You would follow this guide to repair the string properly. Similarly, if a new DNA sequence with regions of homology to the target is provided, HDR uses this as a template for repair of the break. This allows for the integration of new gene sequences into the genome, which we refer to as gene knock-in. So, through these processes, either the gene becomes non-functional, known as gene knockout, or new gene sequences are integrated into the genome, known as gene knock-in. Genome editing, in essence, is a fascinating game of microscopic cut and paste with the potential to rewrite life as we know it. But did you know there are newer forms of gene editing technology that do not induce double-stranded breaks? Instead, they have taken a different approach to rewriting the book of life. These cutting-edge methods utilize either engineered or inactivated nucleases. These are essentially molecular scissors. But instead of cutting both strands of the DNA, they only snip a single strand. Now, this might seem like a small tweak, but it is a significant one. By making a single-strand cut, these techniques reduce the risk of unwanted mutations, a major concern in traditional gene editing methods. But there's more. Some of these advanced gene editing technologies don't cut the DNA at all. Instead, they cleverly identify and bind to the target sequence in the gene. Once attached, they direct a completely different enzyme to perform the actual DNA modification. It's a fascinating world of molecular gymnastics, isn't it? Each gene editing system, whether old or new, has a unique set of advantages and disadvantages. Let's recap what we've learned about genome editing. At its core, this is a process that allows us to change an organism's genetic code with the help of tools called nucleases. These enzymes act on DNA, guided to a specific target site in the genome. Once the target DNA is recognized and bound, the nucleus paves the way for editing through several pathways. When double-stranded breaks are created, the cell tries to mend the break, either through non-homologous end joining, which can lead to gene knockout, or homology-directed repair, which can integrate new gene sequences into the genome. In the realm of new gene editing technology, we're seeing a shift away from inducing double-stranded breaks. Instead, engineered or inactivated nucleuses are being used to cut a single strand of DNA or identify and bind to the target sequence, directing a different enzyme for DNA modification. And that, in a nutshell, is how genome editing works. Isn't science fascinating?